All right, let's get into it. So, broadly, I'm going to be talking about problems related to predicting the function of DNA sequences, particularly with respect to their regulatory properties. How do these sequences influence the regulation of nearby genes? There's a bunch of different ways to justify that. I think it's a fairly basic computational biology problem with a lot of applications. I'm going to give you one here quickly, although it's really not the one that I think Carl's going to be digging into, which is just a sort of more general and quite creative application of these sorts of models. But in my current position, we're very interested in aging-related diseases, common diseases. For the most part, these have very large GWAS now. This plot, which is rising through 2016, has only continued to do so in the time since. So for many of these traits, diseases, we have many associations with the phenotype, each of which is representing a hint about some aspect of genomic function related to that disease. But of course, most of us have seen many of these, a Manhattan plot of the different associations and their p-values. And when you're lucky, you can label it with a gene because maybe you do have a coding variant in there or some sort of other obvious function that ties it to that gene. But more often, you have many different non-coding variants. Many of them are associated with the phenotype because they exist in linkage disequilibrium in the genome, and the target gene and relevant cell types are not at all obvious. So really, that's a big goal of the research field, and any tools that we can build as computational biologists that try to accelerate this process can have a pretty big impact. So one of the goals of this line of research is to build a resource of genetic variant effect predictions and connections between these genetic variants, as well as the loci they exist in, to genes in the genome and, numeric values on those edges that describe their function in different cell types and tissues and situations. Okay, so now to the basic problem here. We would want to be able to predict gene expression and often other attributes of chromatin from the DNA sequence. So we're going to arrange the following machine learning problem. We're going to take a region of the genome, DNA sequence, think at least tens and maybe hundreds and maybe millions of nucleotides as input to this machine learning model. And then we really want to be able to recapitulate all of the existing data that we have for any experiment that someone's done, which you can browse in the genome with a view like this. We want to be able to understand the mapping between DNA and these annotations. And if we do so, I would argue that we understand their regulatory properties pretty well. So the primary tool that I've been using that has been very successful for many over the last few years is deep convolutional neural networks. So the convolutional neural network, in its initial interaction with the input DNA sequence, has these filters that we're scanning across the sequence, which are quite capable of learning the transcription factor binding site motifs that we'd been previously using and representing via other mechanisms. So you have these convolution filters, they are randomly initialized, and then as the model is trained, they're gradually becoming versions that look something like the ones I've drawn here, where they're learning to pick up on different patterns that are useful for making predictions ultimately. And those tend to be TF binding sites that we can then take and go compare to our databases of motifs and label and try to understand. So here, this is just the sort of basic convolution block. We're going to do this many times. We're going to repeat that. And then each time we do it, we're going to use a pooling operation to decrease the size of the sequence. So this has a few different justifications. You're starting with something that's a very large sequence, and you're gradually shrinking it, but each of the vectors that represents one of the individual positions in that sequence is growing in size typically, and it's certainly having to grow in terms of information content, because you're going from initially representing a k-mer to, as you go towards a larger region of sequence, now you have to represent a more complex regulatory element. So at the top here, I've drawn a bunch of them that we really hope these models are able to recognize. We want to be able to pick up on promoters and enhancers and insulator elements and whatever else is out there in the jungle of the genome. So after this stage of having run multiple convolutions over the DNA sequence and trying to learn local sequence information, now we have this task. If we are working in large sequence land, I should say here maybe that a lot of publications, a lot of the models that have been put out there and studied in depth, they kind of stop here after the convolutions. They don't need this other stuff because typically they're working with a shorter sequence, and they're really just trying to predict the local properties of that shorter sequence, such as its accessibility via DNase or a TAC-seq, for example, or a ChIP-seq binding site. But to try to think about gene expression, you have to begin to work with larger regions of sequence and synthesize information across a larger region that can also contain more distal regulatory elements. So the question is, how do you do this? And it's a problem I've been thinking about for, given this publication date, probably 5 years. So the initial solution that I came up with was called Dilated Convolutions. This is a tool that's been used a bunch now in genomics, as well as in image segmentation and lots of other image-based tasks. And the idea is the convolutions have gaps and they are able to reach further. And so you can actually have the region in the center here, which represents a promoter, be seeing a little bit further out so that it can hopefully pick up on these enhancers and other stuff around. All that is now being brought into the vector representation for each position. And then ultimately, in the final layer of the model, we're going to try to make a prediction for lots of different data sets that are available. As much as I could bear to tediously prepare for this machine learning exercise is basically what gets put into this last stage. So here, this is the Genome Browser stage down at the bottom. We're trying to recapitulate that Genome Browser type view in bins of sequence that are, in this case, 128 base pairs. Okay, here is a qualitative example of what that could look like, the experiment, and then a prediction from a model that was trained on regions of the genome separate from this one. And one thing I want to note, even though I'm sort of reviewing an older publication here, this has been true ever since, is that the quality of the training data matters tremendously. If we break down our accuracy on held out test sequences by assay, then first of all, there's quite a difference in the accuracy that you're able to achieve. For something like H3K9-methyl-3, our accuracies are rather low. For something like the CAGE and DNase, they're a little higher. I think the main driver of that is the signal to noise and sequencing depth of the assay. In the case of some of these modifications here in the bottom right, the x-axis is plotting the correlation between two replicate experiments, and you can see that it's quite low in some of these cases, and that really hinders your ability to learn a good model. So just want to note that, because I think as we move forward with this field, one way that we can get better models is follow the latest machine learning conferences and implement the latest exciting algorithms. And the other way we could do it is maybe just redo some of these experiments or bring in better data more generally. Okay, so after this initial publication, we had some tantalizing signals that this was a useful thing to be doing. But nevertheless, the range of interaction for the models was less than satisfying. We were able to capture interactions at about a 20,000 nucleotide range. And if you've studied enhancers at all, you know that there are many, many, many enhancers that exist beyond that distance from the transcription start site of the gene that they regulate. So one of the big challenges we took on moving forward was to try to determine how to increase what we'll call here the visible range of these models. Simultaneously, there was a very exciting development going on in natural language processing machine learning, which was a different type of neural network block, not a convolutional net, not a recurrent net. That had been the focus of most of the research up to this point, but a block that we'll call self-attention. It's also sometimes called the transformer block, but I think the attention terminology is useful, so I'll stick with that for the moment. How it works is that you're coming in with your input representation of a sequence A. So in this case, A is our input. It has a length 3, 1, 2, 3. And each of these is vectors with some depth. So they've done some learning. They're already representing a local amount of sequence. And our challenge is, how do we get them all to communicate with each other so that if there is an important piece of information on the far left and something on the far right really needs to know about it, that there's a mechanism for that to happen. The mathematics of the mechanism is as follows. We're going to do 3 linear transformations of these A vectors into what are called queries, keys, and values. The queries intuitively represent, the keys intuitively represent who am I? What is the information that is contained in that vector? The query represents what am I looking for? So for any one position, it may have a sense for what it wants to look for in the other positions of the sequence, and these query vectors capture that information. And the values specify what do I have to say. So if some other position is going to want to communicate with me, then what is it that I have to tell them? Okay, so these queries and keys are going to interact over here in the upper right to form what's called an attention matrix. There's a few different ways to do this. The simplest is to just take a dot product between each of the keys and queries. And so each of these entries in the attention matrix is a dot product between query I and key J, or vice versa. It should matter. Wait, I should get this right. Query I, key J. I think let's go with that. Sorry if I mix that up. At any rate, each of the, let's see, it would be the columns or rows. Sorry, it's a little early to be thinking through this on the fly. At the end of the day, though, you're going to use these attention numeric values as weights for the value vectors down here. So then you're taking a weighted sum of these value vectors. So, intuitively, the gist of it is that position 2 here now has a vector of how much it wants to pay attention to positions 1, 2, and 3. You're going to normalize that with a softmax so that it sums to 1. And then each of those numbers is going to serve as the coefficients for the value vectors, and then you take a weighted sum. So at the end of the day, each position has had an opportunity to consider the information in every other position, and then extract this weighted sum of information of the values. And what we're hoping for in natural language processing is the ability to take a sentence, to have vectors that are representing the words, and then to be able to identify important relationships in the sentence or paragraph that you would need to influence the next round of processing of that sequence. So for example, the vector representing apple here, it might come in knowing that it's an apple and knowing nothing about what kind of apple it is. And so the attention mechanism is going to allow it to identify the word green. In this case, it's next to the word, so that would have been easy to do with a convolution. But imagine that it was like a full sentence away. She is eating an apple, period. The apple was round and large, comma, it had fallen on the floor, comma, and by the way, it's green. Even in that case, you would have been able to identify green and now modify the vector for the apple to contain that attribute. That's the general idea for NLP. In genomics, we want something somewhat analogous, perhaps. So we have vector representations of now 128 base pair bins across our sequence. So they're roughly representing regulatory elements. So imagine a case where we have a promoter, which has some information contained in its vector. We'll use these colored rectangles to represent something like a transcription factor binding site. So we've already recognized some evidence for the sequence motifs that would determine that. Now we're going to look around and we're going to look upstream a bit. And even though this vector, this position is closer, there's very little interesting content there. So we're going to pay low attention. However, way downstream over here, there's an enhancer that has some other interesting TF binding sites. And even though it's further away, we have this mechanism now to identify that these two positions in the sequence have some information that they would want to share with each other. We can have high attention between the promoter enhancer, and now the next vector representing the promoter can contain some information from that enhancer. Okay, so we put together a model that was sort of a next generation version of the previous one that I showed you, which we'll call Basenji. And it has a numeric 2 because I put out 2 publications improving the model 1 additional time. The new model we'll call the Informer. It became quite trendy in this field to use the suffix former, stolen from the transformer name for the self-attention block that I just described. So we created a portmanteau between enhancer and transformer, informer. Plus, when you say it out loud, it sounds like it might be informing you with an I, which would be nice. That's the general, the etymology of this name. Okay, so it's pretty similar to the previous model initially. We're going to be doing convolutions, nonlinearities, max pooling as before, and we're going to get to this representation of the sequence in 128 base pair bins again. But now, instead of dilated convolutions, which are not an effective way to see at great distances in the sequence, we're going to use these transformer layers instead. The transformer block is typically this self-attention layer that I just described, followed by a feed-forward net to also extract some more useful information. So we're going to repeat that many times so that there are many opportunities for each position in the sequence to look around and extract some new information. And then ultimately, this model is going to be trained on data from 2 different species, human and mouse, which I'll say a little bit more about in a second. But first, I want to highlight collaborators who are real drivers of developing this project. Ziga Avcek at DeepMind and a team of engineers over there was really helpful in getting the self-attention neural nets training. It took a fair number of modifications to what we were doing before to actually get this thing off the ground. And then we made use of some hardware through Google called TPU, which is not critical, but was helpful. And Vikram Agarwal in my group was a major driver too of this research. Okay, this might be a good place to pause and see if there are any questions since I've just been forging right ahead.
That's been a really lucid introduction. I'm just spinning around to see if anybody in the room has any questions. And at the same time, if you're joining on Zoom, you can just unmute yourself and speak up. Share your video if you feel like it. But just so you know, if you share your video or voice, you're agreeing to appearing in the YouTube recording. Okay, seems like there is one question in the room.
Yeah, thanks so far. On the previous slide, I guess a question I had is it's just technical, but about the padding and about how, because if it's 100 kb, that's like a huge, pairwise attention matrix to compute. So how did you guys get it working with that size? And also about the padding on the, so am I correct in understanding that each the length is the same after each of the convolutional layers, or is the length reduced?
The length is reduced. Yeah, that's the main answer to that one. Is that the size has been reduced by 128x because we've done a bunch of max pool operations with width 2 to get it down to where each position is roughly representing 128 base pairs. So now the length of this thing is what? I forget exactly, but it's on the order of 1,000-ish positions, which is now sufficient to represent the quadratic attention matrix. But the point you're making is quite important as we move forward and think, well, wouldn't it be nice to work with larger sequences or to work at finer resolution? And both of those are challenged by the quadratic complexity of the attention matrix. There are many publications now of ideas about how to improve the complexity of the self-attention block. And so this is actually an area of active research in my group to try to figure out which one of those will be the best for us.
Great, thanks a lot.
Okay. I think I saw a hand raise through the Zoom interface.
Yes, could I ask a question still?
Yeah, go ahead.
So here you use for evaluation the Pearson R coefficient. So I wonder if you looked into other ways of evaluating the regression, such as R squared, the coefficient of determination, which is a common metric.
Yeah, we're usually computing both of those. Ultimately, I guess I feel like getting the exact value if you're slightly miscalibrated in some way is not so much an issue, particularly when we're thinking about variant effect prediction and we have a internal control of the two alleles. So that's the main time I see the R and R squareds diverge a little bit, as if sometimes the Pearson R's are high, but because we're doing this gradient descent algorithm, you're kind of swaying left and right a little bit, and it can become the case that your distribution of values becomes slightly miscalibrated. This would be easy to fix, but sometimes you can have the R squareds drop a little. I think it tends to not be a problem.
Thank you.
Okay, going forward. All right, I am gonna actually speed up, I think, a little bit and just maybe sort of skip this. I guess I'll just quickly say that, we're training on human and mouse. That might sound a little weird. Check out this paper if you want a little more convincing. Basically, the way regulatory sequences are interpreted in each genome is sufficiently similar where we don't want the exact same model for both of them. We need some species-specific parameters, but most of the parameters of the convolutions can be shared across mammalian species. And if you train on both species versus one, you can get better accuracy on held-out test sequences. Okay, hopping back to the self-attention. So one thing you may be asking yourself, given the current description that I've given, is how positions in the sequence and self-attention communicate their distance to each other. That, certainly in promoter enhancer biology, the distance that you are away is incredibly important. And so if we just had the self-attention as described, it would be hard to identify if an enhancer was very close by and very likely to regulate, or if it was really far, given the constraints of our sequence length, and maybe we should be downweighting it a little bit. So the initial transformer publication had some fairly simple ideas for how to inject positional information. The gist of it is you would have some complicated function of distance that projects into a vector space, and then you would add that in to each of the positions. But what it ended up being was an absolute positional encoding. So the position I in a sequence of length L would have some information injected that tells it its position I. But because we're randomly sampling sequences from the genome, the absolute position doesn't really matter. What's much more important are the relative distance between two positions. So there's a scheme from a publication called the Transformer XL, and we basically borrowed it with a couple modifications. The general idea is that we have positional encodings for relative distances between positions. So these relative distances are getting mapped through these functions on the left to vector space. So the simplest one that I like is the most is this bottom one, where you basically would have a vector and it's asking the question, is the distance between these two positions less than x? And each position in the vector is an exponentially increasing value of x. So you would have a bunch of ones until you get to the distance where you're now too far, and then a bunch of zeros after that. So, these are getting now added into the representations before the self-attention mechanism computes, and you can actually take the math and manipulate it and sort of split it out. And so it's all done in a reasonably efficient way. But on the right here, we're showing the influence of these different versions of positional encodings. Ultimately, in the publication, we used all of these positional encodings. It's kind of not very harmful to just throw the kitchen sink at it, and you ultimately have parameters that learn from these. And so anything that's important will get used, and anything that's not, or anything that's redundant, just won't get used. But broadly, you can see that these do matter quite a bit. In order to beat the previous model, you really have to get this positional encoding right. And in order to actually make effective use of long range information, you have to get this right. Okay. Another qualitative view into being able to take a region of the genome. Here's a bunch of DNA and chip and now cage tracks down at the bottom for this region. The green track is the actual experimental data itself. The orange track is the previous model using dilated convolutions, and the blue track is the new Informer model, which in this region is producing much stronger fit to the experimental data. Across the genome, that's true as well. So comparing the previous model on the x-axis to the new model on the y-axis. So the plots here, we've broken it down by assay. We have cage, chip for histone marks, chip for TFs, DNase and ATAC-seq. And then each of these dots is one experiment. So on the held out sequences for one experiment of cage, of which there are something like 500 or 600. Here's the correlation on the old model and the correlation on the new. So you see a nice boost, primarily for the cage, and a little bit less so for many of the transcription factors and histone marks and accessibility information. our hypothesis for why that's the case is that CAGE is measuring gene expression. Gene expression is a more complicated function of the sequence, which depends more on these distal enhancers than some of the other tracks do. And so the boost here is fairly consistent across CAGE, and we believe it represents the ability to take into account enhancers across a larger region of sequence. Okay, at the bottom here, we're just analyzing by genes rather than by unbiased genomic position, and we still see a useful boost in accuracy. Okay, I'm gonna skip this. Really, this is just a bunch of sort of hyperparameter scans and ablations of different components of the model to try to convince everyone that what we're doing is useful and necessary. And go right ahead into an application of interest to many, which is the ability to identify promoter-enhancer interactions in the genome. So first question we had for the model was, how is it using this longer range information from enhancers? What ways do we have to interrogate the model's behavior for these longer range sequences? So we did have a hypothesis coming in that the attention matrices themselves would be interesting to look at. And so the track here, the attention track, is showing that. If you take the center position here, which is centered on the transcription start site of this gene, HNRNPA1, then you can extract a 1D signal from the 2D matrix, and this is what it looks like. So you do see that the attention heads, I should say, there are many attention layers. Each of them actually has several independent attention matrices. So this is an aggregation of all those, taking an average, I think. So that is a bit of a challenge. There's actually quite a few, maybe 40 of these attention tracks. But broadly, you can see that there is attention being paid to more distal sequences, not just the local stuff. A somewhat model agnostic statistic that you can compute is to take the gradient of the prediction with respect to either the input nucleotides or some layer of the network that represents more than one nucleotide. Either way, there's a common way to scale these gradients by just taking a dot product with the input vector. So in the case of the nucleotides, this is just saying, what's the gradient to the actual nucleotide? I'm going to throw away the gradients to the alternative 3 nucleotides that are not the reference genome sequence there. And if I then aggregate the statistic in bins, I get the track that follows. So if we do compute this track for the older model and the newer model, you can very clearly see the extension of the visible range that we've achieved. For the dilated convolutions, you don't have any gradient signal beyond the 20 kb-ish range, which is the furthest distance that a dilated convolution is hitting outward in the sequence. But for the self-attention model and former, basically the whole sequence is in play. We have some strong gradient signal for sequences that are quite far away. Okay, so is there any useful information in these tracks? Are the spikes in the gradient track actually representing interesting distal enhancers that we could use this model in order to sort of prioritize them or build out a resource of some sort? That was our next question. And for that, we turned to some validation data, which is based on an experiment where broadly you're looking at a gene, you have some ideas for candidate enhancers in the region of the gene, and you're going to use the CRISPR machinery. So you're going to have a guide RNA that's going to take the CRISPR machinery to this putative candidate enhancer. And then the easiest thing to do is to use something like CRISPR-I, where you're trying to block that regulatory element, because we believe most of these are activators, we believe, blocking them would result in a decrease in expression of the gene. And this has been done with single gene screens by folks at the Broad, including Jesse Ingrates. And in that case, you're doing like a FACS sort on the expression of the gene and then sequencing bins and identifying enrichment of guide RNAs in those bins. Ultimately, you come up with a score for each of these candidate regulatory elements. This has also been done using a single cell RNA-seq readout, where you can come in a little more agnostic to the particular gene you're going to measure. You're going to measure all the genes, and you identify within single cells what was the perturbation that cell received, so that you can break down your cells into cells that received the perturbation and cells that didn't, and do a statistical test. Okay, in both cases, you have lots of labels on putative enhancers, and we can ask the model here to try to predict those labels, or more specifically to sort the candidate enhancers in a way that allows us to compute binary classification statistics like area under the precision recall curve, which is what you're now seeing here at the bottom of this slide. So the data set on the right are the single gene screens from Foucault et al. At the Broad. And on the left, you have the single cell RNA-seq screen, Gasparini et al. From University of Washington. And in both cases, we've taken them and separated them by distance, and we're showing accuracy statistics in these different distance bins. Distance is such an important driver that if you compute your AUPRC across the whole data set, it can be a little harder to interpret. And so, we do see a clear drop off with distance. As you move away, it becomes harder to identify the true enhancers from the ones that land as negatives in this experiment. Broadly, though, you can see that the informer model, of which the gradient by input version seems to be the strongest way to annotate the sequence, is pretty competitive with some simpler baselines, one of which is the old model, which is not very competitive because it can't see very far. And the other is basically different versions of taking into account two pieces of information that we know are very important, which are the distance between the two and the activity of the regulatory element as measured by something like H3K27 acetyl. So that's a pretty strong baseline, and it wins at great distances, particularly in the Foucault et al. Data set, but broadly, and in the single cell data set, we're fairly competitive, which is interesting because, in some sense, either of these could achieve the goal you're hoping for, but the informer model version just offers you a lot more options for things to do. I think Carl's talk to come is going to give you a great example of that. But you can also go in and start dissecting these regulatory elements at a finer view, which we'll do with the last few minutes here. Okay. So I started the talk by arguing that a justification for this line of research was to predict genetic variant effects and hopefully use those to find map and better understand the mechanisms for genome-wide association study associations. So the simplest way to make use of the model to do something like that is just to make a prediction for one allele, make a prediction for the other allele, and compute some summary statistic of the two. The simplest is to take the difference between the two and then just sum across the sequence. So that would be a gene agnostic version of this statistic. Or if you have a gene in mind, you can pick out the transcription start site and just look at the difference in the predictions at that transcription start site. Okay, so here's a genetic variant where we've done that. We saw a high score. We decided to dig in a little bit further. So the tracks here are representing the experimental data for PBMCs using the CAGE assay. So we're seeing very high expression of this gene, NLRC5, from the most upstream transcription start site. The prediction for the reference genome, the major allele from the model, is the following. It easily identifies this transcription start site, as well as this other weird peak down here. And you look at the alternative allele and take the difference between the two, you see that there is some change locally, but the biggest change is upstream here at the transcription start site. So if we come in, this is the genetic variant itself. This C in the reference genome becomes a T. If you go into this region of sequence, you can introduce all possible mutations to each of these positions nearby and make the following sort of visualization where the heat map is showing you the change in the prediction for that nucleotide. And then each of these columns is normalized to sum to 0, sorry. And then the height of the nucleotide here is proportional to its score. So when the allele is C, it participates in this SP1 binding site motif, which helps drive expression of this gene. But when you modify it to a T, it breaks the motif a little bit. The motif is no longer recognized as strongly, and the prediction for the gene is decreased. Okay. So, all right, I think we have time to go through this. So one way to benchmark how well we're doing at generating these variant effect predictions is to make use of EQTLs themselves, which are trying to really give you quite similar information by going out into a human population and just measuring gene expression for a whole bunch of people who have one allele versus another. So the GTEx project is an incredible resource for this. Many different tissues where we have a large enough sample size where we've been able to identify many EQTLs. I was lucky enough to participate in this collaborative project with Hilary Finney-Kane and grad student Qingbo Wang, where they were trying to come up with a prioritization score for EQTLs. And the path to get there was to take fine-mapped EQTLs, so cases where the LD in the region and the data that you've measured is such that you're able to mostly unambiguously identify a single variant as the causal variant. We're going to generate those with a couple of statistical software tools like SUSY or FineMap. And in the latest version of GTEx, there are enough fine-mapped EQTLs to serve as a training data set for like a meta machine learning exercise here. So we're going to take our genetic variants, we're going to label them with everything we can think of, distance to transcription start site, overlap with lots of peak data sets from Encode and all sorts of other resources, and then adding in the predictions from the, in this case, Basenji model. This sort of predated the Informer, but conceptually the exercise here is the same. And, you know, to skip ahead to the point here, we're able to train these meta classifiers for EQTLs. You can distinguish fine-mapped EQTLs from a negative set. And the features from the Basenji model were made use of by the model. And that was useful to Qingbo and Hilary to perform functionally informed fine mapping of those EQTLs. So to take some cases where ambiguity remained as to the causal EQTL, and then resolve a few more of those, and take some cases of large credible sets and shrink the credible set by using functional annotations. Okay, so to sort of skip back to Informer, what we would want to see here is that on this task of EQTL classification, that we're able to improve the accuracy at identifying fine-mapped EQTLs from negative examples. And that's what you're seeing here. Each of the dots is a GTEx tissue. The x-axis is the AUROC at classifying EQTLs with the old model and then the new model. And on the bottom here, you're sort of seeing that again broken down by distance and seeing that really at all distances, performance improved. Arguably, it's a little bigger at the larger distances, as we might have expected. Okay, I'll just sort of quickly scan through this. There's a data set that's really neat, which is a saturation mutagenesis using a parallel reporter assay of 20 different regulatory elements. This was part of a DREAM competition maybe 3 years ago. And this was another way that we benchmarked the model and saw strong performance at predicting mutation effects. Okay, to summarize, so we introduced here this self-attention mechanism, which improves regulatory activity prediction from underlying DNA sequence. It primarily does that by making better use of long-range information, although often the local predictions improve as well. So arguably, either the self-attention is helping that, or by accounting for the long-range information, you're sort of better able to understand what's going on locally as a possibility. And this model is beneficial for variant effect analysis, classifying eQTLs, better matching mutagenesis assays, and so hopefully useful for some of the downstream applications we care about too, which is an active area of research in my group, as you might imagine. And just a couple routes to continued improvement of performance here that I'll mention. Some of these we already talked about a little bit. It would be nice to work with longer sequences. Sequence length here was 200 kb, so you can see left and right, 100 kb for stuff that's in the center of your sequence. We want to go a little further than that. There's enhancers that are beyond that distance, and so we'd like to capture them all. There are some publications out there that have argued for working at finer resolution, which are very compelling, particularly when you have the training data to support it, like high resolution chip assays like ChipXO. So we'd love to be able to take advantage of that great data. Of course, single cell genomics is a huge growing area still. And most of the results I've shown you are from data generated by consortium projects 5 to 10 years ago, which were all using bulk assays. So we'd love to be able to make use of single cell versions of these profiles that go from, a kidney track to tracks for each of the dozen or more specific cell types in the kidney. And then, at the bottom, some crazier ideas I have to work with even more mammalian genomes to give us more training data. Okay, with that, I'd like to thank Zika and many collaborators at DeepMind, Vikram and Han in my group at Calico, who contributed to the project. Mostly Vikram contributed to this project, but everybody's chiming in on everything. And Qingbo and Hillary as well for really interesting perspective on eQTLs and a really great collaboration.
Yeah, thanks for the introduction. Can everyone hear me okay? Mic is working? Great. Yeah, so I felt I needed to include inference in the title, given that it's an MIA talk. We're not going to talk about math or algorithms, but there's a lot of inference today. The first project I'm going to talk about was actually started while I was a postdoc here at the Broad Institute. And I've since moved to the University of British Columbia, where I'm continuing on some of the same lines of work, as you'll see. So just a brief overview. The problem we're addressing today is trying to understand how genome regulation evolved. And the approaches we're going to use are to measure the activity of naive DNA. And by naive DNA, I specifically mean DNA that's never had the chance to evolve in a cell. So the other approach we're going to use is to use gene regulatory models to infer how these sequences are evolving. And just a brief look at what we're going to show. So we found that selection on promoter gene expression is, you can detect it using these models, and it looks like it's under stabilizing selection as expected. We found little evidence for selection on the complexity of the regulatory solutions that the cells are coming up with, which I'll explain in more detail. And very interesting finding recently is that biochemical activity is not a marker of function, which I'll get into in the later half of the talk. So thanks again to David for the great introduction. So he talked a lot about why we care about the DNA sequence. It's what dictates gene expression largely. It's what makes your blood cells and not brain cells. And it's also the underlying cause of many diseases, as Dave mentioned. So the language of DNA was introduced by David, but I'll just briefly go over this. It's made out of the 4 bases, A, T, G, and C. And the words that the language of DNA uses are kind of like transcription factor binding sites. The grammar is incompletely known despite many decades of research. We understand a lot about some of the mechanisms that are happening in the cell, but predicting it is still an ongoing challenge, as David described. So we know that transcription factors can modify expression directly. They can also modify chromatin and modify expression through chromatin. They can also modify each other, so either inhibiting each other's binding or promoting each other's binding. So how complex is this problem? If we just consider one of the parameters we want to learn, so this is cooperativity between pairs of transcription factors, there are a very large number of possible ways they can interact, and as a result, a very large number of parameters to learn. So the equation for how you would calculate this is here. And humans have about 1,640 transcription factors. So if we just consider all pairwise interactions between them, there's over 300 million parameters we want to learn. Very large number. And the number of regulatory regions we have to learn this is on the order of like 50 to 100,000 per cell. So we're pretty underpowered. So in comes yeast to the rescue, except it's not really rescuing us all the way because there's fewer transcription factors in yeast, about 209 or so, which means that the number of parameters we have to learn is much less. But now we're even worse off with the regulatory regions. There's only like 6,000 in yeast. There's no enhancers in yeast, and the promoters are relatively short. There's only about 6,000 genes, so about 6,000 regulatory regions. So in either case, we have too few examples of regulatory DNA to learn all the parameters we want to learn. So we recognized a possible solution to this in the form of random DNA. And we found that if you just inspect the motifs for transcription factors, you see the title is slightly hidden there, but hopefully you can appreciate it anyway. So if you just inspect these motifs and you calculate how often you expect to see them in random DNA, most of them are actually quite common. So this is the equation for figuring out how often you expect to see these motifs. So this is a relatively information-rich motif. Transcription factor's name is REB1. And even this one you expect to see about once every 16,000 bases by chance in random DNA. Most transcription factors are more like risk-free here with a much lower information content. Here you expect to see it about every 100 base pairs. And if you just add it all up across all the different transcription factors, you'll see that the average 80 base random sequence, which is what we're going to use in these experiments, is predicted to contain about 138 binding sites for 68 different transcription factors. So just by chance, you're going to get a very large number of potential transcription factor binding sites. Now, this is in theory. We wanted to make sure this was actually true. So what we did was clone in a random 80 base pair sequence in this promoter-like context in front of a yellow fluorescent protein reporter, and just like took several clones from this plate so that each of these clones has only one promoter sequence, and compared that to the library, which has a diverse set of sequences. And you can see that each of these random 80 base pairs encodes for a specific expression level. And the library overall has very diverse expression levels, which is totally consistent with these sequences being filled with transcription factor binding sites. So this led us to believe that random DNA is going to be ideal for learning this sequence to expression function because the sequences are unrelated. So it's trivial to split it into training and test sequence sets with no risk of leakage. As I just showed, there's probably diverse transcription factor binding sites, including strength, position, orientation, and many examples. So as you'll see in the next slide, we can quantify the expression of hundreds of millions of these sequences. So how do we quantify it? So same promoter context as before, random 80 base pairs embedded in a promoter-like sequence, controlling YFP. Here, we're actually sorting, in this case, by the ratio of YFP to RFP. Same vector has an RFP, but it's under constant control. And this is just to control for things like cell size. Yeah. Put it over there, wherever that is. No, thank you. Thank you for solving that problem. Yeah, so the RFP is just controlling for extrinsic sources of variation that are not due to the promoter sequence. So you can see it's got the diverse expression as I showed before. So how are we going to measure it? We can actually sort these cells using cell sorting, fluorescence activated cell sorting into different expression bins. And if we regrow this, we can see that you get a nice ladder of expression levels, indicating that we're getting a reproducible expression measurement. So instead of doing that, we can actually isolate DNA from all the cells in each sorting bin and sequence it. So in the end, this gives us a gigantic table with expression levels in one column and the promoter DNA sequence in the other column. And the DNA sequence in this case is exactly what we read off of the sequence. We don't have a reference set to refer to because we don't even know what sequences were in the library until the very end of the experiment when we sequenced them. Using this approach, you can get about 30,000 different sequences per experiment, which is equivalent to a human genome's worth of DNA. But this is being measured in yeast. And to date, we've measured, this number keeps growing. This is out of date, but it's well over 15 gigabases of new DNA measured today. So now we have the perfect data set for learning this sequence to expression function. Now I'm going to talk about the model that was created. So actually there's two models, one of which, the first one that we made is an interpretable model. It's explicitly trying to include parameters for transcription factors that we know are present in yeast and learn what they're doing, how they're binding DNA, how they're changing expression. Its power is that it's interpretable and it has pretty good predictive power, but it's not as good as the neural network that Ishit Davo-Vaishnav, who was a PhD student in the Regev lab, who's since graduated, although I hear he's sometimes in town. He had a conference, so he couldn't be here today, but his neural networks were really excellent predictive power, but not as easy to interpret. So we use them instead for inferring how mutations in the sequence can alter gene expression. So what do I mean by excellent predictive power? Here I'm showing the predicted on the x-axis and measured expression on the y-axis for different sequence sets, including 10,000 random promoter sequences. This is again measured in the same system we used to measure the training data, but a distinct set of sequences. And 5,000 sequences from the yeast genome. These are like parts of the actual yeast promoters measured again in this system. You can see that in either case, it's explaining quite a lot of the variation in expression. So now we're going to use Ishit's model to, as basically an oracle to understand how these regulatory sequences evolve. We know that it works very well, and I'm not going to talk about all the validations we did, but there are a very large number of validations. So we know it works very well at predicting expression from sequence, and we don't care how it gets the answer, we just care that it gets a pretty good answer. And now we're going to explore how sequences evolve using this paradigm. And we're not the first to do this. So there are many other groups, including from David Kelley, where people have used deep learning or other machine learning models to understand the effects of variance. And we're building on that. So the first question we're going to ask is, can we quantify the selection on promoter sequences using the deep learning oracle? So first we need some genetic variation data. And actually, the human genetic variation data is now much better than yeast, but yeast, we still have some pretty good data sets, including this one, where they sequenced 1,011 whole genomes of Saccharomyces cerevisiae isolates. So we took these data, and for each promoter, we identified all the orthologous promoters amongst these isolates and cataloged the genetic variation in that promoter. And then we create a matched set of simulated variation where the distances are preserved, but the mutations are now randomly placed throughout the sequence as like a null hypothesis. So then we calculate the standard deviation of the predicted expression levels for both the simulated set of variation and the actual observed set of variation. And the intuitive understanding here is that if there's a lot more greater standard deviation in the simulated variation than the actual variation, it must be because these are under selective constraints. The ones that deviated too much from the desirable expression level, those yeast died. So that would result in a positive, calling it the expression conservation coefficient. So sure enough, you look across all yeast genes and you see that the expression conservation coefficient is shifted to the right, consistent with conservation of expression. So these ones are more conserved, these ones are more divergent in their expression level. And if we look at the bottom end, the genes that have the most divergent expression level, they are enriched. It's not like a very significant enrichment, but these are nonetheless genes that are involved in carboxylic acid and alcohol metabolism. And if you think about where these yeast strains were isolated from, it's basically diverse fermentable habitats across the world. So it's really not surprising that they would have to adapt alcohol metabolism to these different environments.
Quick clarification. So this is conservation of the promoter sequence of the gene, not the gene sequence, or?
Yes, this is only based on promoter sequences. And on this end, these are the ones that are not conserved. These are the most divergent. So this could indicate local adaptation of the fermentation gene and all these other genes to the local niche of that isolate of yeast. On the other side, we have these core cellular processes like RNA and protein metabolism. This is not surprising because if you mess up the ribosome, then the yeast is dead. So these ones are highly conserved. So it's not that there's no variation in these genes. In fact, we see variation. The variation just preserves the expression level. So one interesting example we see, yeah, sorry, was there a question on Zoom?
Yeah. Hey, Carl. Can you hear me? Just a quick question. It seems to me like your the focus on variance reduction rather than a mean shift. Maybe there's a piece there I'm not understanding, but that would mean that you're focusing on stabilizing selection rather than sort of positive or negative selection. And it makes a lot of sense in this context, but I was wondering if you agree with that or if there's something there that I misunderstood.
No, I don't think you misunderstood anything. So a mean shift would also be interesting, but it can be difficult to know where it's coming from and where it's going to. It's not like we have an ancestral sequence that's clear, and we can see that over time the mean is shifting from one to another, indicating like adaptation. But instead, we just have like a snapshot of the variation in the population. So it's hard to know if it was like coming from somewhere and going somewhere else as like a mean shifting over the generation. And actually, if you look in detail at the paper, you probably wouldn't expect to see that anyway, because a gradual shift, it turns out you can change expression, at least of yeast promoters, very quickly with 1 or up to 3 mutations. So rather than seeing a gradual shift over time, you would see a single mutation potentially altering the expression level of the promoter substantially. Does that answer your question?
Yeah, that's great.
Okay. So this one promoter we saw looked very interesting to us, CDC36. So this is a gene involved in the cell cycle, specifically involved in mRNA turnover. That's not super important to what I'm going to show you. In this promoter, it was very interesting in part because the model predicted it came in 2 expression flavors, high expression and low expression, and not really anywhere in between. And using the biochemical, the interpretable model, we asked it what was going on, and it said it's really changing a binding site for 1 transcription factor, mostly. It's UPC2 transcription factor. And here I've included the motif here. And you can see that actually this expression shift evolved twice independently, which we thought very interesting. One of them is this very conserved C to a T in this lineage of yeast. And in this other lineage of yeast, which expanded very substantially, it was a G mutating to an A. So in both cases, it's mutating these critical bases. And actually this one strain here mutated the second base, the C to a T, which is also an important but less so base. So it looks like a case of convergent evolution by mutating the same binding site for the same transcription factor and resulting in the same change in expression. We wanted to validate this. This was a lot of work. This was done by Jennifer Molinet in Francisco Cudios's group. He's at U Santiago de Chile. And what they did was take a strain that lacked the binding site. So this is one that had lost the binding site and restored it base by base. It actually had the 2 mutations in the binding site. Restored one, restored the other until it was the ancestral UPC2 binding site, the consensus UPC2 binding site. So this is what the predicted expression looked like for these alleles. And here they measured it by qPCR, and you can see they correspond very closely to each other. It's really remarkable how closely they matched. And they were even able to find a phenotype for these yeast. So as I mentioned, the gene is involved in mRNA turnover. So we hypothesized that this might enable the yeast to adapt to changing environments by changing how quickly the transcriptome is turning over. And sure enough, when you switch carbon sources, they can adapt more quickly with the higher expression of CDC36. So that was a pretty cool example where we had a model that predicted the effect of a variant. And then it took a lot of work to actually validate that model's prediction. So if there's any questions about this part, now would be a good time because we're going to talk about something slightly different.
The model predicts.
Yeah, so what is the range of
Repeat the question, please.
of the model? So it's kind of arbitrary units because it's based on cell sorting data, but we have calibrated with natural sequences, and it seems to be at least within the kind of promoter scaffold that we have in the system. Natural sequences don't have greater variation than random, so it looks like the random and the model predictions as a result are spanning pretty much the full range of expression. There's like some edge cases on the very high end where it becomes difficult to measure, and also at the very low end. But it looks like they're not substantially different from actual sequences from the genome. All right, so the next question we're going to tackle is whether selection shapes the complexity of regulation or just the expression level. So in order to answer this, we need a way of quantifying the complexity of a regulatory sequence. So we're doing this using the Interpretable Model. And as I said, this one is explicitly modeling how each transcription factor is regulating the DNA. So we can mess with the parameters. We can say, okay, we predict expression of the sequence with the full model. Now do the same thing, but delete one of the transcription factors. Now what do you expect is going to happen? And now we can use the predicted change in expression as kind of an interaction strength between the transcription factor and the promoter sequence. And we can repeat this for all transcription factors, creating this regulatory network for a sequence and how it is regulated by all the different transcription factors. So here are two examples of promoters. And these ones were selected because they actually have the exact same, within the precision we can measure anyway, expression level, but they have very different regulatory complexity. And in this case, complexity, we're quantifying it by 1 minus the Gini coefficient of all these interaction strengths. And the Gini is just a measure of inequality. That's why we're doing 1 minus it. So in this case, this is a low complexity sequence where one transcription factor is contributing pretty substantially to the expression of the promoter. And in this case, it's a high complexity sequence because many transcription factors are contributing much more equally to its expression. So now we're going to see whether this is an important parameter in yeast promoter. The way we did this is by starting with a set of either high complexity sequences. These are from the yeast genome, yeast promoter sequences that had a high regulatory complexity or a low regulatory complexity. And we simulated stabilizing selection. So we take this sequence, we generate all possible mutations. We use Ishut's model to predict the expression level of all of these. And then we select one, but we select one that preserves the original expression level. So it's a neutral mutation, thinking that these other ones, if they perturb the expression level by more than a threshold, and threshold turns out doesn't matter that much, if we perturb expression, those yeast died. So we're going to not select that mutation. So we just keep doing this. And then eventually we get this trajectory of mutations, increasing numbers of mutations, but the expression level is pretty well constant. And we're going to see what happens to regulatory complexity as this happens. And you can see that the high complexity goes much more moderate as we increase the number of mutations. Same with the low complexity. This is moderating, so it's increasing in complexity in that case. So if it started low, it can go high, and if it started high, it can go lower. And these were selected by being the extremes of this natural distribution. But what this is telling us is that the sequences, if all we care about is expression level, there's no reason they have to be high or low. It's more like they just happened to be high and low. Something had to be the highest, something had to be the lowest. We picked those at the beginning of this experiment. And you can maintain the expression level while perturbing the regulatory networks. And the other important finding here is that if you look at those that are from the genome and those are random sequences, they don't really look substantially different, indicating that the regulatory complexity of sequences in the genome is not substantially different than if you were just doing random walks with selection on gene expression. You're just sampling solutions from the space of possibilities, and when you get the right expression level, you stick with that solution. The regulatory complexity itself doesn't matter, at least for most genes. In some cases, I'm sure it matters substantially, but most genes, it doesn't seem to matter. Okay, so this is the first part. The gene expression oracle we found can identify expression evolution using population genetics data. We found this really interesting example, CBC36, but there's little evidence for selection on the complexity of regulation. Now, this paper is a very dense paper. We had a lot of things going on. I didn't talk about most of it, but if that piqued your interest, you should check it out. And since then, we've been further developing these models. This is spearheaded by a graduate student in my lab, Abdul Muntakim Rafi. Ishit did a great job with optimizing the neural network that he built, but he's only one person. So we knew we could do better. We held this dream challenge to predict expression using these random promoter expression measurements. And it's since ended. The top performers were these teams. And the important bit is that they really kicked our butt. They did an excellent job, made much better models than we had done previously. So this is great. It's exactly what we wanted to happen. So this is with respect to both models, but the deep learning model was better than the interpretable model, so especially compared to the interpretable model. But the only solutions submitted really were deep learning type models. So we're going to present this in November at the RSG Dream. So if you want to see more, we'll have it out soon. So next, we're going to talk about how we found that biochemical activity appears to be the default state of DNA. So this was a project started by a postdoc, Cassandra Jensen, in the lab, and she did the experimental stuff in yeast that I'm going to talk about. And the computational experiments were done by a PhD student, Ishika Luthra, and a summer student who's now at UPenn, Amelia Chen. So let me just remind you, we saw a lot of striking similarities between the genomic regulation and random DNA in the first part. So we saw that random DNA could encode for diverse expression levels, just like the sequences in the genome. We saw that the regulatory complexity of the sequences in the genome is pretty close to the same as what you see in random sequences. And although many of the sequences appear to have conserved expression level, there's actually a lot of sequences near zero here, which is consistent with random drift of the expression level. So just some caveats. This is based on mostly results from the computer model. We validated it extensively, but it's still a computer model. Some of these things could be wrong, and observations from a reporter system. So maybe this is not a general case. But keep in mind here that so yeast has, because of its relatively large population size, about 1,000 times the sensitivity to selection as humans, which means that something that causes a fitness defect in humans has to be 1,000 times the fitness defect for us to efficiently select for it. This has to do primarily with the fact that throughout most of our evolution, our population size is quite small. So if we're seeing all this activity of seemingly random sequences in yeast, what is it going to look like in humans where the selection is so different and the genome size is so much larger. So there's ongoing controversy over the degree of adaptation and function in the genome. So for instance, in 2012, Encode famously said that 80% of the genome had biochemical function. Now, what does the biochemical do in that sentence? I'm not entirely sure, but since then there's been this raging debate of how much the genome is functional. And the latest estimates are less than 25%, probably much less than 25%, probably less than 10%. And this is in part based on the fact that we can't get rid of deleterious mutations fast enough if too much of the genome were functional. So nowhere has this been more hotly debated than with link RNAs. So here there are about 10,000 to 100,000, depending on how you ask, link RNAs. There are some that we know have a really important critical function to the cell and the organism. Many others remain uncharacterized. Now, are they simply just uncharacterized? We haven't gotten to them yet, or are they like noise in the system? So in 2013, in response to the Encode paper, Sean Eddy proposed essentially the random Encode project. So suppose we put a few million bases of entirely random synthetic DNA into a human cell and do an Encode project on it. Will it be reproducibly transcribed into mRNA-like transcripts, reproducibly bound by DNA binding proteins and reproducibly wrapped around histones marked by specific chromatin modifications? I think yes. So we wanted to test this. So I showed you the reporter data where we had random DNA changing expression in yeast. Turns out other people have done similar things in humans, and there too it appears to have functioned random DNA. This recent paper by Sahu et al. Was particularly interesting. So the goal here is to make a draft random Encode project for yeast and humans. And the idea here is that we're going to have the evolved DNA, the DNA that evolved in that organism, and compare that to naive DNA, DNA that did not evolve in that organism, and compare what we see the cell doing with it, or what computer models that predict biochemical activity from sequence think is going to happen with those sequences. So we're going to start talking about yeast. This is all going to be experimental data. We're going to use something called a yeast artificial chromosome for this. And these were used for sequencing the human genome. They're about 100 megabase in size. It's human DNA. It's already in yeast, so we didn't even have to make this. They existed already. Now, human and yeast, we're both eukaryotes, but there's a billion years of separation, evolutionary distance between us. So we are quite distantly related. The transcription factors bind DNA and activate expression in similar ways, but the transcription factors are almost entirely different. And the gene structure, importantly, is very different. Yeast genes are tiny and have few introns. Human genes are huge and have many introns. So first we needed some YACs. And in an interaction that I think looks something like this, Cassandra contacted Steve Scher's group at Sick Kids Hospital in Toronto. His group was responsible for human chromosome 7 for the human genome sequencing project. And they used YACs extensively in sequencing the human genome. And amazingly, they still had some 30-year-old YACs in the freezer. So that really updated my prior for how much smallpox is still in the world, but was very convenient for us. So in the end, we got 10 YACs, but we only actually used one because doing more was not going to change the answer we were seeing, as I'll show you in a second. We performed strand-specific poly-A plus RNA-seq and aligned to a hybrid genome comprised of the yeast genome and human chromosome 7. And in the analysis, for everything I'm going to show you, we're comparing expression from evolved sequences to naive sequences. So it's always very apples to apples. We're ignoring any genome annotations there might be because those would bias our results. So we're just comparing. We do all the analysis and then just split it up to from where it came. So the first result is that naive DNA is really transcribed very extensively. And that's shown here. So this is the Rnac coverage. This is a section of the yeast genome from this experiment and a section from the human genome, chromosome 7, same experiment. And you can see coverage is shown in blue for the top strand and red for the bottom strand. And then we have also predicted transcripts from cufflinks and annotated genes for the yeast genome on the top one and human genes on the bottom one. You can see that, sure enough, the genes look like they're transcribed in yeast. In this case, this is a human promoter, but it doesn't appear to be a promoter in yeast. It's actually in the middle of the transcript. And in the human DNA in yeast, it's being transcribed all over the place. Some of the naive transcripts appear to be spliced. So here's an example where there's actually 4 different isoforms of this transcript. They are using the canonical splice motifs of yeast, which are similar to the ones in humans, but in this case, it's a transcript that is not, as far as I know, it doesn't exist in humans. It's being transcribed in yeast. And fortuitously, these splicing signals are there. Yeast is using them because it doesn't know that they're not a real gene. So some of them are spliced. So just some like accounting of what we're seeing here. Overall, we have 760 kilobases of naive DNA, which compares to the yeast genome of about 13 megabases. The transcript density is pretty similar, so the number of transcripts per kb. And overall, about half as many of the transcripts are spliced in the naive DNA as the evolved DNA. But the transcript length is more or less the same. It's actually slightly longer for naive transcripts. They're both about 2 kb long. The expression levels are also pretty comparable. But in this case, there's a long tail of high expression in the evolved sequences, indicating that you really do need selection to achieve these really high expression levels. So rather than relying on the ability to call transcripts from these data, we next just looked at a per base coverage. So comparing the reads on strand 1 and the reads on strand 2 to see how much transcription is happening on both strands. And since this would be a symmetric graph, we've just like put evolved DNA in one half and naive DNA in the other half. And you can see that evolved DNA has much less antisense transcription, so much less DNA that's transcribed on both strands. It also, evolved DNA has much more DNA that's not expressed at all, which you can't really see here because it's so small down in the corner. But about 3.4% of bases have no expression from the yeast genome, only 0.6% of bases in the naive genome have no expression. So you can see that here looking across these two things, many of the yeast genes have no antisense or very little antisense expression. In this case, this GAL1, this is actually a known regulatory antisense transcript. So this is kind of exceptional, this locus, whereas in the human DNA, it's transcribed on both strands all over the place. And very few of the bases here have no expression, but you can see here's an example, and here are some examples where there's no expression in the yeast genome. So the yeast genome appears to have optimized this in a way that it's only expressing specific regions and a more coherent gene structure than would be expected if it was just random DNA. So now we're going to try humans, and doing the same experiment in humans would be really hard. So we can't really do it. And the experiment we could do there isn't really adequate anyway, because even if you could synthesize 100 kb, which is possible now, it would take you a long time to do it. But even if you could synthesize 100 kb of naive DNA and put it in a human cell, you only expect about 0.6 genes there. So you put it in the cell, you see a gene that might tell you something. You see no gene, that basically tells you nothing. So it's really not feasible to test this in the same way in human cells. So that's why we're going to use Informer, and I'm not going to talk any more about this. Hopefully, David Kelley convinced you that this model is good. And the only thing to note is that we're only considering the sequences that they retained in the test set. So these are sequences that were not used to train Informer, which is important. Make sure we're not like overfitting to these sequences. So the first result we had is that random DNA appears to be regulatorily silent in according to Informer. So here we're showing one particular locus. These are the genes. These are the different chromatin marks predicted by Informer. This is for embryonic stem cells, the H1 cell line. And so this is the same tracks, but for a random sequence of DNA. You can see there's not much going on here. So random DNA appears to have almost no activity in human cells, according to Informer, but the human genome is not random, not even close. It has massive changes in the base content. In particular, if we just consider CpGs and GPCs, GPCs outnumber CpGs by like 3 or 4 to 1. So the dinucleotide content is heavily skewed. And if all you're doing is recapitulating the mononucleotide content, then it doesn't look anything like the human genome. So now we tried different versions of this randomization, including global and local shuffling of the sequences and preserving mono, di, and trinucleotide content. And you can see that completely random or mono or di or tri at a global scale, all of these result in very little activity. But once you start going into local shuffling of dinucleotide content or trinucleotide content, you've restored a lot of the activity. So it's not that this naive DNA had no activity in the human cells, but it didn't look anything like the human genome. So if you just preserve local shifts in dinucleotide content or trinucleotide content, which is basically the same thing, but more, then you preserve this expression of the sequences. So this is just showing a cumulative distribution plot where we have the cumulative proportion here and the DNAs, predicted DNAs on the x-axis, and you can see, we'll zoom in on the top corner here because otherwise you can't see anything. And usually we assume that somewhere around 99% of the genome is regulatory, so it should be accessible by DNA. So we're going to say, okay, this is going to be the cutoff. Everything above this, we're going to call a regulatory sequence. And we're going to say, okay, anything that has this predicted level of DNAs or higher, we're going to say that is also a predicted regulatory sequence. And we can see that about 0.6% or about 60% of the activity of the human genome is predicted to be present in these dinucleotide, locally dinucleotide or trinucleotide shuffled sequences. So they do appear to have some activity. So henceforth, I'm going to use naive DNA to be locally dinucleotide shuffled genomic DNA. So just to make one important point here, when you do this local dinucleotide shuffling, it's not simply maintaining the activity that was already there. So this is shown here for 3 different cell types. Each of these dots is one of the Informer bins, the predicted 128 base pair bins, and they're like the same genomic locus, except in the naive, it's been locally shuffled for dinucleotide content. So you can see that they don't, the sequences that were active initially after shuffling are often inactive, and the sequences that were inactive before shuffling can become active. Interestingly, the naive DNA is more cell type specific than the evolved DNA. So here I'm showing this is for evolved, for the 3 different cell types. So these are the 3 different germ layers, just representing very diverse cell types from humans. And you can see they're pretty tightly correlated. This is DNase 1 again, and much less correlated for the naive DNA, indicating that it's more cell type specific in its activity than the genomic DNA. So now this will be the last analysis I'm showing. So Hopefully we can end close to on time. That should be okay. You're probably used to seeing these piece-knee plots in the context of single-cell genomics, but in this case, each of these dots is a 128 base pair bin from one of 100 loci that we included in this analysis. And they either originated from evolved sequences from the genome or naive sequences, which are the dinucleotide shuffled evolved sequences. And here we're clustering this based on these chromatin marks, in particular, using the data from H1 embryonic stem cells. So yeah, they form this nice, pretty hairball. So now we're gonna look at the individual histone. Excuse me. Here I've split the data up again into evolved sequences on the left, naive sequences on the right. And I hope you can see, it's actually pretty hard on this projector, but okay. So you'll just have to believe me. We're looking at DNAs now, and you see some sequences that have DNAs over here, and they have DNAs over here. There's more over here, and some over here and here. And basically the gist of this is that it's been clustered by these chromatin marks, and you're seeing similar things in both of them, in naive and evolved. And so this is now a H3K4 trimethylation, which is canonically associated with promoter activity. And we see the same thing in naive and evolved. And this is H3K4 monomethylation, which is associated with gene bodies and also enhancers. And again, we see similar sequences in both. And this one's kind of a mystery to us still. This is like hot off the press, but this one is confusing because you can kind of see it. It looks like there's just overall much more predicted histone K27 acetylation, which is associated with promoter and enhancer activity in naive DNA versus evolved. It appears to be more localized to specific regions in evolved, but more everywhere in naive. And like I said, I don't really understand what's happening there. There's also H3K27 methylation, which is associated with repression. And that is also present in both. So the point of this is that both in naive and evolved DNA, you're seeing this co-occurrence of these chromatin marks, indicating that the fact that they co-occur is not a sign that those sequences evolved, because that's just how the system works. So conclusion for this part, we created a functional genomics null hypothesis. We found that naive DNA is biochemically active in yeast and humans, and it looks a lot like the evolved DNA in many respects. So biochemical activity by itself is not a marker of function. And organisms appear to just have evolved to do genome-y things when you give them DNA, because they're not used to having random DNA put into their cells. So important caveat, the human analysis I showed is all based on a computer model, but we trust it for this kind of analysis. So one last thing, I showed you this experiment here, where we're looking at the variant effects on expression. As David mentioned, we're also very interested in this problem, and the predictions of the model were very easy. And in this case, the experiment was very hard. Same thing is true in humans. It'll take you like 3 years to validate SNP function in humans, but the models are getting better and better, and we're hoping to create a system that will allow us to predict variant effects and also design regulatory sequences across all cell types of the human body. And the way we're doing this is by engineering this reporter system in pluripotent stem cells. And the idea here is we now have this tool that will allow us to downstream create these gene expression models using high throughput reporter measurements. And because we've done it in pluripotent stem cells, we can differentiate it to whatever cell we want, and then create the appropriate data, create the models, predict how to interpret these regulatory variants, and how to design sequences. So let me know if you're interested in helping with this, because there's too many cell types of humans and we are not an expert really in any of them. So we need help characterizing all these cells and figuring out how we can predict variant effects across all tissues. With that, I need to acknowledge Cassandra Jensen, who's now at Boreal Genomics. She was a postdoc, Ishika Luthra, and Amelia Chen. This is for the second half of the talk of the naive versus random DNA. Aviv Regev, of course, who's moved from Broad to Genentech. I was in her lab while I was a postdoc here, working with Ishik Devil Vaishnav and Esteban Luis Beta. He helped with some of the experiments, and of course, Francisco Tridios and Jennifer Molinet, who did that allele swap experiment in yeast. And with that, I would be happy to take any more questions you might have. Thanks.